Welcome everyone to your own channel, ADP for Learning. I am your own learning partner cum host, Ashish. So, let's begin our learning journey. Today, we are continuing our learning journey about the Interactive Glossary of Genomic and Genetic terms. And our motto is, 5 terms at a time, keep the learning in the mind. The first term of today is carrier screening. Carrier screening involves testing to see if a person carries a genetic variation or allele associated with a specific disease or trait. A carrier has inherited a normal and a variant allele for a disease or trait-associated gene, one from each parent. Most typically, carrier screening is performed to look for recessively inherited diseases when the suspected carrier has no symptoms of the disease. But that person's offspring could have the disease if the other parent is a carrier of a harmful variant in the same gene. Carrier screening is often thought about around the time a person may have a child, such as when a person is considering becoming pregnant. The person, sometimes with their partner, undergoes carrier screening to see if there's a chance that their child could be affected by a genetic condition. There are also other times that carrier screening may be done. Carrier screening is a great reminder of the way in which genetics is truly a family affair. That is, it shows how genetic changes can be shared among relatives and can be passed from one generation to the next. The second term is popular with several names such as cDNA or copy DNA or complementary DNA. cDNA, short for copy DNA, also called complementary DNA, is synthetic DNA that has been transcribed from a specific mRNA through a reaction using the enzyme reverse transcriptase. While DNA is composed of both coding and non-coding sequences, cDNA contains only coding sequences. Scientists often synthesize and use cDNA as a tool in gene cloning and other research experiments. cDNA, copy DNA. If you get to spend a week in a genomics lab, it is very likely that you will hear about cDNA one way or another. That's how important this word is, and for a good reason. Imagine that for one of your experiments, you need to make a protein and a lot of it. Let's say you want to create a more stable version of insulin, a diabetes drug. One way to do it is to get cells that make this protein, let's say pancreatic cells. These cells will contain a gene with instructions on how to make insulin. These cells can also transcribe this gene to temporarily store this information in the form of RNA. Using a special enzyme called reverse transcriptase, you can convert RNA back to DNA, but the resulting DNA will look a little different from the original gene. For example, it will be much shorter than the gene from where it came from. This is why scientists use another word, cDNA, copy or complementary DNA, to tell these versions apart. What you can do with the cDNA of insulin is very interesting. You can clean it up. You can make changes to the sequence. And using special molecular tools, special enzymes, you can insert it back into genome of another organism, for example, yeast. Yeast is very good at making lots of proteins. And if you insert cDNA of insulin into its genome in the right place and configuration, it can start making insulin for you. This is one way scientists can make and test new versions of insulin with properties that can be helpful in treating diabetes. The third popular term is cell-free DNA testing. Cell-free DNA testing is a laboratory method that involves analyzing free, that is, non-cellular DNA contained within a biological sample, most often to look for genomic variants associated with a hereditary or genetic disorder. For example, prenatal cell-free DNA testing is a non-invasive method used during pregnancy that examines the fetal DNA that is naturally present in the maternal bloodstream. Cell-free DNA testing is also used for the detection and characterization of some cancers and to monitor cancer therapy. Cell-free DNA testing was something that really changed many areas of clinical medicine and continues to do so. It's still a very active area of research in terms of ways to look for different types of genetic changes and in different clinical areas. Cell-free DNA testing is a great example of how genetic discoveries translate to different fields of medicine, such as the way cell-free fetal DNA is used in obstetrics practice. The fourth term is centimorgan or cm. A centimorgan, also abbreviated as cm, is a unit of measure for the frequency of genetic recombination. 1 centimorgan is equal to a 1% chance that 2 markers on a chromosome will become separated from one another due to a recombination event during meiosis, which occurs during the formation of egg and sperm cells. On average, 1 centimorgan corresponds to roughly 1 million base pairs in the human genome. People can think of a centimorgan as a unit of distance. Yet, in the strictest sense, it's not really. It's a measurement, or rate of how often something occurs. In this case, recombination or the exchange of DNA during meiosis. The greater the distance between two genetic markers, let's say it's two genes, the higher number of physical opportunities for the exchange of DNA to occur. So you have a higher frequency of recombination and a higher number of centimorgans between the genes. Fun fact: centimorgan is named after Thomas Hunt Morgan, an American geneticist who worked on fruit flies, a commonly used model organism for genetic research. The fifth most popular term of today's video is central dogma. The central dogma of molecular biology is a theory stating that genetic information flows only in one direction, from DNA to RNA to protein, or RNA directly to protein. The fundamental theory of central dogma was developed by Francis Crick in 1958. His version was a bit more global and included the notion that information does not flow from proteins to nucleic acids. Scientists have since discovered several exceptions to the theory. A particularly notable example is that of prions. Prions are infectious proteins which replicate without going through DNA or RNA intermediates. Prions are responsible for the rare but devastating neurologic disease Creutzfeldt-Jakob, which is a uniformly lethal disease that causes degeneration of the nervous system. Thank you for joining with me for the amazing journey of learning. In the future, we will again meet with other new, innovative topics. Meanwhile, don't forget to like, share, subscribe, and comment.